non-native protein structure that alters the protein function and then subsequently promotes the aggregation. We also know that at least about 4 different scar types are caused by large non-protein coding repeat expansions, which bind to and sequester RNA binding proteins within the intranuclear RNA, which results to either RNA toxicity or splicing. And in the cytoplasm, we can see expanded repeats that can engender repeat association using the non-ATG mediated translation, which can lead to aggregate-prone polypeptides. And so these RNA translation may even occur with some of the expanded CAG repeats, which are underlying the polyglutamine diseases. What we also know is that many scars are in fact directly imposed by mutations in genes that encode the ion channels or the ion channel associated regulatory proteins that lead to ion channel dysfunction. And other types, particularly the polyglutamine scars, they are indirectly associated with changes to the channel function or expression. So as you can see, there are multiple ways within which this disease mechanism can be occurred. So if we look at the history of the SCAR genes and then the types of mutations that one find, within the gene structure, we have repeat expansions that can occur in either the 5' UTR region or the 3' UTR regions, which have specific scar types that they cause by presence of mutations in specific genes. For example, the SCAR12 subtype is caused by a mutation in the PPP2 R2B gene. We also have repeat expansions in exonic regions as well as intronic regions. So as you can see, repeat expansion can be very critical to detect, particularly when you're doing molecular diagnostics. And then we have also conventional variants that includes, for example, the missense variants, the nonsense variants, or the deletions and the insertions. And so the multiple genes have already been reported to cause a specific SCAR subtype. And so all of these come into play when we design specific panels to do molecular diagnostics. When we talk about the grouping of these variants, you can group the repeat expansion into the CAG polyQ scars, or you can even have a group with the other expansions that have specific sequences that cause a specific scar subtype. We also know from the scars that you have non-repeat mutations in certain genes that cause the multiple scar subtypes, as you can see in the group from the third group. And sometimes we also don't know which gene and which mutation is caused by that particular phenotype. When we look at the molecular genetic diagnostics of SCAR as a flowchart in routine clinical implementation, when you have clinical evidence for SCAR, for example, you have progressive ataxia or a similar disorder in the generation, or no evidence of an acquired cause, so you know that this is probably hereditary. And then if the SCAR genotype is known or highly suggestive phenotype is available, then you probably want to go for a targeted gene test, which when turns out negative, you can then go for a translated CAG repeat mutations in specific SCAR genes. And so that becomes a decision that requires a multi-group discussion, whether it is the lab technician, whether it is the counselor, whether it is the medical doctor, or whether it is the bioinformatician, looking at what would be the most appropriate first site of investigation when looking at these mutations. When your translated CAG repeat mutations panel turns out negative, you can then look for untranslated repeat mutations, for which already SCAR genes are known. And if that also does not work, you can then look for whole exome or whole sequencing to look for specific mutations. So as you can see, because of the heterogeneity of the phenotype and the heterogeneity of the genotype, it is a very complex process within the clinic, but also in the diagnostic, the routine implementation on how we can find these mutations that will explain their phenotypes. So when we talk of patients which probably did not receive a diagnosis, so an undiagnosed patient, what can that be done to such a patient? So in this scenario, the patient might have a conventional mutation in a non-coding region of a known scar gene. That could be one alternative, or a mutation in an unknown scar gene. That could be another alternative as well. But sometimes, and this is where I will really focus in today's talk, something called the variant of unknown significance in specifically known scar genes, which has a significance. So the scar gene is already known to be implicated to cause the phenotype, but then the mutation does not have sufficient evidence to be classified either benign or pathogenic, and therefore it remains classified as a variant of unknown significance. And this is what we will focus on today. But the problem is that studies in affected patients that use next generation sequencing based approaches for the conventional variant detection, such as the missense, nonsense, and insertions and deletions, certainly leads to a notable increase in the diagnostic rate. We've seen in the clinic multiple cases whereby patients do benefit from further investigation. But the problem is that, utilizing the next generation sequencing panels, a lot of variants get classified as variants of unknown significance, which then becomes a burden because you do not know how to classify them, number one, and number two, they are not clinically actionable. And so that becomes a challenge in the clinic. And so really, this is a question whereby we want to be able to generate enough evidence that allows us to classify a particular variant of unknown significance as benign, which means it is not disease-causing, or as pathogenic, which means it is disease-causing. Consequently, when we talk about the clinical practice of how to deal with the variants of unknown significance, I think everybody probably recognizes this chart that when you have a variant, the classification using the ACMG guidelines can bring it down to 5 main groups. So you have the benign, likely pathogenic, and the variant of unknown significance as the key groups that these variants can be classified and put under. And so the burden really is the variant of unknown significance, and one way to solve or to reduce that burden by providing additional evidence such that these variants become clinically actionable. We want to supplement these particular variants by either looking at family studies, so we really want to understand if there is any parent or grandparent that has the same mutation that might explain the phenotype. So we do family studies, or the alternative way would be to be able to do a functional study. And so that in that way, we analyze the mutation and the gene, particularly in the lab, whereby we want to understand if there is any changes to the expression, particularly on those particular variants, so we can get additional evidence to make the classification. Now, what we have done in this particular study is that we address the challenge of prioritizing the variants of unknown significance. Particularly, we wanted to follow up those that are more likely for us to be able to get additional evidence in the context of getting a scar genetic diagnostics. And I will explain to you in the next slide how we make these groups and how the functional testing was then subsequently done. So we also present a procedure, which is a new way to prioritize these variants of unknown significance to do a functional follow-up. And the functional follow-up can be done using aggregation analysis, as I mentioned, using family history and family information. We can also do a protein modeling, and we can do a splice assay. So this is really using the mini gene assays to detect an expression difference for splicing variation, as well as cellular testing by looking at the translocations that may be present when we do cellular assays. So in our cohort, we had a total of 368 undiagnosed Dutch scar patients. When I say undiagnosed, this means that they have already been through a fully Q-tail assessment to understand if there is any repeat expansion. And so once we, in our clinic, in the diagnostic setting, we first find out if any of these patients carry a poly Q repeat expansion, and if most of these patients, when they do, we at least know that that particular mutation might explain their phenotype. However, when the test results are negative, we then try and look for conventional variants. And so in this particular study, we have designed a new targeted gene panel that covers 36 SCA genes. So these are the 36 genes that have been indicated to cause a SCA phenotype. And we try and look at all the exons of these 36 SCA genes together with at least about 25 base pair of the intron, positive and negative intron regions for each of those exons. So we have a bit of intronic information, but also a lot of this information is exonic. And essentially what we are trying to look for are the conventional variants, as I mentioned, because these patients did not, they tested negative for the repeat exposures. What we found when we look at the results is that we found a total of 330 unique conventional variants from these 300 plus patients. And we used a software called Alamut. Alamut is a software that is a very easy to use tool for lab technicians and molecular geneticists to understand the amino acid chemistry as well as the amino acid sequence homology and information on splicing, because it provides multiple algorithms to be able to make a decision on whether or not we want to classify a certain variant based on the in silico evidence that we have. And using Alamut, we've classified together with the ACMG guidelines these variants in-house using our diagnostic decision tree. And so what you can appreciate on the pie chart on the right is that of the variants that we found, about 203 were likely benign. We found more than 20 that explains the phenotype. That means they were either likely pathogenic or pathogenic. But as I mentioned earlier, the huge burden of these variants are variants of unknown sequence. And so the 106 in the purple section represents the variants that, in fact, are at the moment clinically inactionable. And what we wanted to try and do is see if we can further utilize functional testing and family segregation data to narrow down some of these variants. So in our publication, we've provided an extensive list of the pathogenic and likely pathogenic variants that we've identified in more than 20 patients. And as you can see clearly, most of these, the typical candidates pop up. So that includes the CACNA1A gene, the KCNC3, the KCND3, which explain the SCA6, 13, 19, and 22 subtypes. And so these are all provided in our data sets. Now, when we talk of the variants of unknown significant subclassification, as I mentioned, Alamut is a software that provides multiple in silico algorithm data for us to be able to get evidence using the ACMG guidelines on whether or not a variant is interesting for a functional study. So we wanted to subdivide the buses, the 106 buses that we found in our data by looking at three main areas. So we wanted to know a pathogenicity score. So this is basically done from 6 main predictors that we use on our algorithm. So this is typically your SIFT, polygen, the align GBHD, the mutation taster. Basically, these are all the typical software that are used to understand the conservativeness of the variant, as well as whether a particular variant is deleterious or it is benign. And what we've done here is that we've identified variants that would increase the threshold of 3. So of the 6 predictors, if 3 or more say that this variant is deleterious or it is pathogenicity causing, then we put this variant under a larger scrutiny where we want to do a functional test. The other parameter that we really bank on is the population frequency. So if we look at the minor allele frequency from the genome data or the exome data data sets that are present, we want to filter out any of those mutations that are rather common, and that means that 0.1% would be our cutoff. So anything below that would be interesting for us, and anything above that would be something that we would not be interested in. And the third group is the splicing prediction group. So Alamut provides 4 main splicing predictors. So this is the NN splice, you have the splice max, and multiple other algorithms that provide a score difference between your wild type and your reference and your mutant. And so if we find that there is a significant difference between your wild type and the mutant for these in silico splicing algorithms, we record these scores. And if 3 out of the 4 splicing predictors have indicated a significant difference, then we want to prioritize this particular variant for a functional splicing testing. So of the 106 buses that you find, we've grouped them into bus A, B, C, and D. So the A means that the pathogenicity score is less than 3, which has a very high minor allele frequency. So that is automatically inclined towards likely benign, so that's not very important in terms of functional follow-up. But it is important when we talk about the significance of a particular variant when we find it. We had the first B group, which means that the pathogenicity score is equal or more than 3, and then the minor allele frequency is less than 0.1%. So this is very interesting to do a protein modeling assay. We have the first C. Sometimes you have variants that are actually not yet reported into these genetic data sets. So this is basically those variants that have very high pathogenicity score using in silico programs, but have no reported they have not been reported in any of the data sets. And finally, we have the first B group, which means that these are groups that have been predicted by the splicing algorithms to have a splicing effect. Now, what does the distribution of these first subclasses look like in our patients is that, when we do the additional sequencing of our patients, we solved at least 6.6% of our cohort. That means 6.6% of these patients, we were able to tell them that the variant that they were carrying is in fact explaining their phenotype. But of the variants that were of unknown significance, we have about 40 patients, which is about 11% that we've identified from the 106 variants that we would like to do a functional follow-up, such that we want to reduce that burden of the variants that have not found an evidence to be reclassified yet. And again, in our publication, we've provided extensively all the list of buses, whether they have been classified as a semi-high or high, or a splice in the list of the patients that we have. So you can refer to this data set whenever you want. Now, the distribution of the variant, which we find in the TAR gene, of the 36 genes, we found 24 genes have majority of the variants that we were able to get a diagnosis for. And like you can see in the chart, that majority of the variants in class B was found from the FAT1 gene. First C was found in the CACNA1A gene, which is again typical of the spinocerebellar ataxia. And the first D was a bit more sporadic in the PDYN, NOP56, FAT1, and CACNA1G genes, which are also very, very important when detecting these splice mutations. So what we've done then is of the 106 variant, sometimes Alamut, you can argue that this is an in silico program, right? And what Alamut does is that it looks at amino acid sequences and it looks at amino acid chemistry. But it really lacks, or it really is limited when we talk about the 3D structure of the protein. And so it is very important to understand the context of the variant in terms of how it affects protein modeling, or how it affects the hydrophilic and hydrophobic bonds that make up the stoichiometric chemistry of these bonds in the protein models. So we identified some of the variants that we wanted to follow up either using protein modeling or using a splicing assay using mini-gene assays. So we have about 20 variants that belong to class B, 13 variants in class C, and about 5 variants in class D. To be able to do so, it is very important that protein modeling can be utilized using X-ray crystallography data. So for a lot of these genes where an X-ray crystallography data is already present, that means the protein has extensively been studied, it is important for us to then understand how these variants would affect the bonds, the hydrophilic and hydrophobic bonds of these particular protein models. At the same time, we also use a Swiss model to define specific homologies for those regions that were not yet done X-ray crystallography on, so that at least we know using a 40% cutoff that the sequences that are provided by the Swiss model would be able to replicate the model that has already been X-ray crystallography, that has already undergone the crystallography, so that we are able to understand also how these mutations would affect the bonds. And so to give you an example, this is an example of a mutation on PRKC gene, and this is at a position 715 that changes from C to T. And subsequently, that means that the protein will change from arginine to tryptophan. And as you can see in the picture on the left, in the wild type, when the original variant is present in the non-patient, you can see that there are multiple bonds that connect the arginine to serine and arginine to glucose. However, gluten. However, in the mutant, the change of arginine to tryptophan all of a sudden breaks out the bonds that would connect the tryptophan to serine and tryptophan to gluten. And so you can already appreciate from these pictures that there is some modification happening by presence of the mutant variant in this cow phenotype. And when we look at the hydrophobicity, in this particular picture, the red bonds represent the hydrophobic regions and the white bonds represent the hydrophilic regions. The original variant had a hydrophilic region, but all of a sudden now the mutation that has changed the protein from arginine to tryptophan has converted the hydrophilic region into a hydrophobic region. And so that has an influence also on how this protein will behave downstream. Using that protein modeling process, we were able to do protein modeling for 8 mutations, of which 4 of them were found to have a pathogenic effect. And these 4 are provided in the slide that you see here. One of them is the R239W, the second one is the LF529P, we have the V1890D, and we have the R212Q. All these particular protein models that we tested variants on showed some sort of hydrophilic-hydrophobic bond interaction change, and then subsequently this has an influence on how the protein behaves such that it is then causative of certain disorders, and then that is the additional evidence that we would require from a functional assay to be able to reclassify some of these variants from FUS to pathogen. When we talk about the predicted effect of the group of FUS-B with the splicing assay, we use the PSPL3 vector, which is basically a plasmid, in which you can easily clone in your wild-type exon, or you can clone in your mutant exon, which has the mutation, and then look at the expression by transfecting these in the cells. We extract RNA, we do cDNA synthesis, and then we do cDNA amplification and check it either on gel electrophoresis and then later confirm it on a Sanger sequence. So the results from the splicing assay you can clearly appreciate here is that for these 2 variants that we found, first of all in the group A, which is the variant present in CACNA1G, we find that the mutation that has the variant had an exon 19 skipping. At the same time, the mutation in the second variant, which is the 909G, had a specific intron 6 30 base pair retention. So depending on the type of splice variation, you have multiple ways of how this DNA makes changes. You have either regions that are increasing the intronic space or skipping some of these exons that have an influence on how the protein function will affect. At the same time, we also did cellular studies in which we found specific PMA-induced translocations were found in mutants. For example, in picture A, we used immunocytochemistry to find translocation that was observed in the wild type but was not observed in the mutant. And then in the second particular variant that we tested, there was no difference between the wild type and the mutant. So collectively, utilizing protein modeling, utilizing splicing assays, utilizing functional testing using cellular assays, as well as family information, we were able to identify certain mutations which will benefit from reclassification from a variant of unknown significance into a likely pathogenic or pathogenic. And so to conclude, I want to tell you that our study showed that reclassification of variants of unknown significance is certainly very difficult due to the limited opportunities to carry out variant segregation studies. As you can see in our study, only 2 variants you are able to find a segregation with a family member. And obviously, the limited weight and availability of protein modeling and functional tests can be also a bottleneck. We also anticipate that this situation is not going to change in the near future, especially because the ACMG guidelines are very stringent, and most of the diagnostic labs lack a protein modeling expert, and functional tests are not easily available. So to solve this issue, what we advise is you should focus first on the most clinically relevant genes, and you try and gain expertise on the structure of these proteins. And it is very important that protein modeling experts play a role in diagnostics, and so that you make functional testing part of your route in diagnostics. And to improve the yield further, the variants of unknown significance that are available clinically functional protein modeling should be widely shared between labs. That is very, very pertinent because then we are able to build sufficient knowledge to weigh the value of the outcomes of these variants in classification, particularly by providing additional evidences and give them more position in the classification guidelines. That becomes very, very important, especially when we talk about data sharing between labs here in Africa, or even data sharing labs between European and African cohorts. So data sharing can be very, very important. So with that, I really want to thank you for your time, your attention. This work was done at the genetics department of UMCG in Groningen, the Netherlands. I want to thank specifically Fatima Gorbani and the entire GCC team, and the drug design and protein modeling work was helped by Matthew Groves and Juliana Bilata. And of course, the most important stakeholder here is the patient groups. So thank you so much.
Thank you, Dr. Mohammed, for that very elegant talk. Now, if there are any questions from the audience, we can take them now.
Okay, thank you for that presentation, Dr. Mohammed. And it seems to me that the Alamut algorithm is like a composite of other algorithms, particularly in silico algorithms for pathogenicity prediction. And we have seen a lot of these composites like meta-LR, meta-SVL, and so on and so forth. So I would like to know that what makes Alamut unique for bioinformatics analysis, especially for diagnosis uses. I can see you made some highlight on it, but I can't really understand what is the unique, what separates it from other composite algorithms that are out there. And then again, the second question is that what is the visual interface like for Alamut? Is it window-based or Unix command lines and so on and so forth for accessibility by people, different people with different skills in bioinformatics? Thank you.
Thank you so much for that question, and I think it is a very relevant question. I think this is something that we really need to think how to bring these kinds of tools here in Africa. Alamut basically is a platform that allows multiple algorithms to come into one unique software. You do not necessarily need extensive bioinformatics training to be able to judge whether a variant is pathogenic or benign for that matter. Because what Alamut does is that it takes all the algorithms from literature that have been known that provide additional evidences to classify these variants into one software. So think of it like it's a platform where multiple algorithms can be embedded into one program, and that program utilizes the different transcripts of a specific gene, and it uses mainstream algorithms to provide information. So Alamut itself does not do any analysis. What it does is it brings in multiple tools in one platform so that you don't have to do complex bioinformatics. It provides you already with the tools in one platform, utilizing existing bioinformatic platforms on one chart. And so when you, for example, when you use Alamut and you put in one particular variant in the software, it already utilizes existing algorithms to tell you whether that mutation is conserved, whether that mutation already has a difference between the wild type and the mutant as far as splicing is concerned, whether there is any deleterious prediction from SIFT or polygen, which are mainstream tools to identify specific protein changes that might have an influence on the classification. So I think personally, Alamut is a very important tool when we come to sort of provide that additional evidence needed for a variant, particularly when we are looking at the ACMG guidelines, because that is one, it's like a one-stop shop for everything.
Anyone else? Well, in that case, thank you very much, Dr. Mohammed, for your presentation.
Thank you so much.
Sorry, there's one more question, please.
Yeah, sure.
Thank you. Good morning, Dr. Al-Zahir. I just wanted a clarification. I hope you're hearing me.
Yes.
About the genetic makeup, of in the African population. If you could comment on the genetic profiles that you gave us in the Dutch population. How about in African population? Do we have any studies showing what are the distribution of these genes and the frequency of the mutant apparent gene that is important in this particular population? If you could just comment on it.
Thank you so much for that wonderful, wonderful question. And I'm really glad that you bring it up, because this is exactly what we need to do now. Because as you probably know, most of these genetic data sets are quite Eurocentric in nature. That means a lot of this information is based on European cohorts. Unfortunately, for the different scar types that I mentioned, there is not much information from African populations, but we can already speculate, given that how different the African genetic makeup is, that the same variants might not necessarily come up when we compare them to European ancestry. And so in my talk, in my next talk, after a couple of other people speaking, I will also specifically touch on the genetics of African populations in the context of pharmacogenetics. And I think for the purpose of the Spinocerebellar ataxia, we really need to find a consortium that is going to look at sequencing and screening patients from African populations to be able to understand the genetic epidemiology, as well as to be able to understand the types of variations that will be present in these scar patients, but at the same time also understand the genotype-phenotype correlation, particularly for these specific subgroups. So I bank on a lot of people that are sitting in the audience to come up with novel ways for us to come together and be able to sequence our patients.
Any other question? Well, in that case, we thank you very much, Dr. Mohammed, for your presentation. And we'll move immediately to the next speaker, who is Dr. Margaret Oluka from the University of Nairobi. within the School of Pharmacy. Please welcome.
Good morning. Morning. I know people now have a difficulty transitioning from virtual to physical until you don't know which one you prefer. Which one do you prefer? Maybe I should go and sit somewhere. But at least I'm here and I'm glad to be here this morning. Just a minute.
Minutes she wrote her presentation to screen.
Good morning. Is it working?
Yes.
Morning. So, my name is Margaret Oluka. I come from the University of Nairobi School of Pharmacy, and I'm glad to be here this morning. Start by thanking Dr. Ngala and the team at IPR for inviting me to this very exciting meeting, loaded with a lot of science, and I'm really glad to be part of it. Next, I wish to appreciate the variety of participation that I've just seen in the audience, coming from different countries, and I think as Kenya and IPR, we should be very happy to see that we are able to attract this kind of attention and participation. So again, those who are not from Kenya, I wish to welcome you to Kenya. And I'm hoping you enjoy yourself in the bush. The road is as bushy as the environment. I think it's meant to be that way. I know I was not aware that there are many students from the University of Nairobi. May I see the participants from UIN in the audience? Carry up your hands. Well, thank you. You're just, I think there were more than that. I was not aware that you're inviting students, especially undergraduates, but I've seen them here and I'm very happy that they are here. And then, of course, I have Benedict there, my PhD student. Since yesterday, he's been here. He's learning a lot. And at the end, he will probably find a very good collaboration in this group. Yeah, thank you. So I know the students from UN are very comfortable because their pharmacology is very solid, what we teach them. So the topic that I was given, well, as you can see from the program, says pharmacology stroke pharmacognosy in Parkinson's disease. So I tried to find something to present in this area, considering the fact that the very next presentation actually is a lot of pharmacology and pharmacotherapeutics. So I tried to find a way in which I'm not duplicating that, the next, or preempting the next presentation. So I've tried to confine my presentation mainly to pharmacognosy, which is a science which is concerned with natural medicines, natural therapy. And that is going to be the focus of my presentation. So I'm going to, hoping at the end of the presentation, I would have explained the pharmacological basis of how natural medicines actually intervene at the pathology of this particular disease, Parkinson's disease. Next. So in the presentation, I'm going to introduce the topic, what is Parkinson's disease, but that one I saw from yesterday, you had a very, very powerful presentations on that, so I will not go there much, but I will give some information on pathogenesis of the disease, the treatment options, and the place we have for the natural herbal products. Next. So in my introduction, I had to put the topic into context, always for us to know what disease we are talking about. So Parkinson's disease was thoroughly explained to you yesterday's presentations. That is a neurodegenerative disorder in the particular part of the brain where there's loss of dopaminergic neurons. But apart from dopaminergic neurons, we also know about the cholinergic neurons. We have the serotonergic neurons. We also have the adrenergic neurons, which are also involved in the pathology of this disease. So those ones, I will not go into that today. Next. Next, please. Yeah, so in the pathogenesis of Parkinson's disease, the progressive degeneration of dopaminergic neurons found in the nigrostriatal pathway There are about 7 pathogenic pathways that are important that I wish to highlight in this presentation, and that is the ones I've listed there: apoptosis, immunological mechanisms related to the disease, the proteolytic defects, the oxidative stress, mitochondrial dysfunction, iron metabolism disorders, and protein misfolding. Okay, before I listened to before I saw the topics that we were doing yesterday, I'd gone ahead in my presentation to try and explain each and every one of these mechanisms of pathology in the disease. But this morning I removed that part because there's really no need of going into details because that has already been explained in another lecture. So I'm using this pathology or pathogenesis really because the medicines that we are using for treatment really intervene at the pathological pathogenesis and pathophysiology of the disease. As those students of pharmacology know, that normally pharmacology is actually a science that is very much related to pathology, because when you know the pathology of the disease, then you are able to design a medicine to intervene at a particular pathway of the disease. Next. So again, I think this is very familiar to you from just to show the particular area where we have the lesion that leads to Parkinson's disease. Next. So this diagram here shows us the various pathological, the 7 pathological areas that I've which I've just listed: the apoptosis, the iron metabolism, the oxidative stress, mitochondrial, protein. So this is just to show us in the middle, there we have the degeneration of the neurostriatal pathway neurons that leads to Parkinson's disease, and how these 7 pathological pathways lead or are linked to the pathology of Parkinson's disease. Next. So this is just again to explain like oxidative stress being one of the pathological pathways found attributed to causing Parkinson's disease, just showing the various ways in which oxidative stress could be generated and its impact in the particular lesion, the impact on the lesion, and how that leads to Parkinsonism. Next. So having looked at the pathology, and now we are aware of the various pathological pathways that lead to Parkinsonism disease, what are the treatment options that we have for intervening in this disease? So treatment options that are available, and I don't want to preempt the next lecture, we have the drug therapy, where we have the conventional drug therapies that are actually given to ameliorate and improve the deficiencies, the various transmitters that are deficient. Then we have the complementary drugs, which are highlighted in red because that is what I'm going to look at in this lecture. Then, of course, the other interventions like surgical therapies, physical therapies, occupational therapy, speech therapy, and others which are not listed that have their own place in management of Parkinson's disease. Next. So I'm going to look at the place of natural products, that is pharmacognosy, the science of how we harness natural products to use them in therapy. So I'm going to look at the use of herbal products in Parkinson's disease. So there's concerted research that is being carried out to investigate the role of natural products in Parkinson's disease. And this is because the conventional therapies have their disadvantages. and therefore we're always looking for better medicines, and therefore herbal medicines are one of those alternatives, particularly because conventional therapies are associated with high incidence of side effects. For example, the dopaminergic based therapies, they cause a condition known as dyskinesia, which is really affects quite about 20% of patients, occurring very, very frequently. And then, apart from the side effects, there is tolerance or loss of response that sets in very, very quick, very, very soon after exposure to these drugs. So therefore, it means that alternative therapy, particularly the natural therapies, have their own place in intervention, in pharmacotherapeutic interventions. Okay? So I'm going to take you through some of the common herbal products that I was able to find that are being used in the treatment and management of Parkinson's disease. So I've selected some herbal products that are very, very common. Most of them are in our houses, in our dinner tables. So you may be able to actually recognize them already, like Macuna puriens, Vicia foba, Nigella sativa, Crotus sativus, and Curcuma longa. These are very common herbs. I couldn't find their Kiswahili names, but I will endeavor to find, and also their local names. So these are some of the common herbal products that are being used in Parkinson's disease. Next. So if you remember this slide, I had placed it there in the pathogenesis of Parkinson's disease. So what I've done is to superimpose the herbs on the pathogenesis of Parkinson's disease so that you can see at what level each one of these herbs are intervening, what is their mechanism of intervention, what is their mechanism of action in alleviation of the pathology of Parkinson's disease. So you have the oxidative stress and a group of herbs that are used to alleviate oxidative stress. You have mitochondrial dysfunction, you have apoptosis, you also have immunological reactions. So you have all these herbs that are able to be used to alleviate these conditions. So next. So like Macuna puriens, that is how it looks like, both the shrub and the seeds that are being used. Next. So this is usually, it is actually one of the Indian traditional medicine herbs. And the main thing important about it is that one of the main active principles is L-DOPA. L-DOPA, as you may remember, is key to Parkinson's disease. It is in the pathway of the pathology. Dopamine is in the pathway of the pathology of Parkinson's disease, the major pathology, and L-DOPA is a major intervention, replacing dopamine. And therefore, this medicine, the main reason why it has found its place in alleviation of the symptoms associated with Parkinson's disease is because it contains L-DOPA. So it is being used in the treatment, and when people use this medicine, it has been observed that there is remarkable elevation of endogenous levels of dopamine, norepinephrine, serotonin, the the substantial nigra. And this is normally observed in these cell lines which are a model for Parkinson's disease. So this work has been done and has been proven that this herb has a place in the management of Parkinson's disease. Next. Then you have the Vicia foba. Again, you can see this is beans. It's a bean, what is available for use. You have the leaves, the stem, but you also have the leaves that are actually cooked and eaten. Next. And of course, known as, its other names are broad beans, and it is used extensively, especially in the East. And again, the main reason why it is used is that one of the major principal components of this herb is L-dopa. So again, it contains a very useful, it has a useful place in the management of Parkinson's disease. And again, studies have been done to show that use of this herb leads to increased concentrations of dopamine and other neurotransmitters associated with the pathology of Parkinson's disease, and of course improvement of symptoms like motor symptoms. Next. Then we have Nigella sativa. Next. Again, the active product known as TQ has been proven to be neuroprotective again when it is used in the laboratory in cells which are models for Parkinson's disease. It has been shown that it is able to cause to provide neuroprotection. So there are two active principles, TK and cavacol, which are major active principles in Nigella sativa, and again, research has been done to prove that they have a very useful effect on Parkinson's disease. They are also able to attenuate neurotoxicity caused by 6-hydroxy dopamine in the Hemi-Parkinson rat model. So again, this has been proven and actually published work to show the importance of these herbal products in the management of Parkinson's disease. Next. So this other one is known as Croton sativus. Next. Again, it contains two active principles, the one known as crocin and safranol, and these are also very important, and also research has shown that they are very useful. They have proven to have a positive effect on Parkinson's disease. They have a protective effect on dopaminergic cells, again, in the mouse model of Parkinson's disease. Next, you have our good friend, which all of us should be able to recognize, Katima longa, the common turmeric, a very useful herb. Next, yeah, it contains curcumin, which is a major active ingredient, which has also been extensively studied and has been shown to inhibit, for example, the monoamine oxidase B activity. It also significantly inhibits dopamine metabolizing enzymes. So the pharmacology has been done and has been proven that the active principles found in these herbs actually have relevant and direct impact on the pathological pathway of Parkinson's disease. Next. So this is just a summary I made to show some of the studies that have been done on the plants themselves, the model, whether it is rat or mice or cell lines, and then the effect that has been observed in the laboratory and what is published in terms of, this is just some examples, but there's plenty of published work to show the role of these herbs in the management of Parkinson's disease. So these are just a list of herbal products that I had put together. These are very common, and actually these can be incorporated in diet because we can use them in our own houses, in our own cooking, to intervene with our patients. Next. Then from now onwards, I have two other important herbal or phytochemicals, medicinal herbs that I'm going to concentrate on. One of them is most important, Withenia somnifera. That's how it looks like. The leaves, the stem, the root. That is the root. Then you have the seeds, and all these parts are very useful. All of them can be used: the root, the leaves, and the seeds. Next. So Withenia, which is common, it is one of the herbs commonly used, which has been extensively studied to be important in the treatment of many other diseases. It is particularly extensively used in Indian medicine, known as Ayurveda. It grows commonly in India, Middle East, and also indigenous in African areas. So it has been shown to exhibit numerous properties, including antioxidant activity, memory-enhancing activity, and also anti-inflammatory effects that have a positive impact on Parkinson's disease. Next. So this is just to show again the plant itself, the neuroprotective effect that it has, not just on Parkinson's disease, but on other neurodegenerative disorders that you can identify with, including Parkinson's disease, as well as aging, where it can be used. Next. So it's phytochemistry. It is, as you can see, I don't know if it is clear, but you can see it contains quite a wide array of active principles. You have from the flavonoids, alkaloids, you have the steroidal lactone steroids, you have the salts, and you have the N-containing compounds. All those are important active principles found in Withenia somnifera, which have been extensively studied and experimentally proven to have an impact on Parkinson's disease. Next. So I've just listed this because it was not there. Maybe the other one was not very clear, but you can see the phytochemicals and the nature. So you have alkaloids, you have steroidal lactones, so you have midaferin, you have midanolides, you have tropin, you have quercetin, you have this diosgenin, and you have withanol. So all these compounds, plus others which are not listed. This plant is one of the richest plants containing a repertoire of active principles that are actively being studied now for purposes of drug discovery and applications in Parkinson's disease. Next. So I just want to take you through one or two studies that have been done in Withenia somnifera. Unfortunately, I was not able to find any local studies to present today, as opposed to the immediate previous presenter was presenting local study that's been done in their laboratory. I didn't get any local studies. In our lab, we do a lot of herbal work, herbal research, but we focus a lot on infectious diseases, particularly malaria, and not neurodegenerative disorders. So this is a study that I found that I thought I could share about the neuroprotective effects of cutaneous omniferra, on the Parkinson cell module, which you can be able to recognize. This is a recent study published last year. Next. So the study objectives were to measure oxidative response, to find out if the active principles, actually they were using the root extract, and they set out to find out if the root extract exhibit oxidative, can be able to alleviate oxidative stress, which you'll remember or recall is one of the 7 or so pathological areas in Parkinson's disease. Also to elucidate the neuroprotective mechanism by measuring percent cell survival, and also looking at some enzymes like glutathione antioxidant enzyme activity, thiol transferase, and peroxidase activities. And then number 3 is to determine expression of oxidative response proteins, again, and to determine the extent of protein glutathionylation. So these were the objectives this paper was addressing. Next. So experimental, the researchers set out to set up various experiments, the determination of the inhibitory concentration, 50 of the test compound, which is named KSM, and the 6-hydroxydopamine, which is the one that is being used to cause or to induce Parkinson's cells, induce Parkinson's dopaminergic deficiency in the cells. Then they set out to do analysis of neuroprotective properties. They did cell treatment and protein preparations for enzyme activity. They were able to determine enzyme activities. They carried out Western blotting, protein glutathionylation, and they were able also to quantify ATP levels in these cells, in these cell lines. Next. So what results did they get? They were able to, so you can see the control is there, and then this is when they treated the cell line using 6-hydroxydopamine to demonstrate that it is cytotoxic, that is the substance that is inducing the pathology in the cells. They were able to demonstrate that it has a dose response in terms of its cytotoxic effect, increasing from small quantities to high quantities. Next. So this is the contrast when you use the KSM66, that is now the root extract, treated on using the same cells. You can see that the picture is totally different compared. It does not show cytotoxicity to the extent that is seen with the 6-hydroxydopamine. Next. This one, the picture we have here is to show the morphological changes associated with treatment of those cells using 6-hydroxydopamine compared to the control. So the control you can see, and then you can see the morphological changes associated with the use of the 6-hydroxydopamine, which is our pathological state, to induce the pathological state. Next. Again, then in this experiment, they had done pre-treatment of the cells within a root extract before applying 6-hydroxydopamine. And you can see that if you compare this with the previous, you can see that these are much lighter. The impact, the cells are much lighter, and of course the improvement in the pathology, in the morphology associated with increased concentrations. So what this is showing is that pre-treatment of the cells with the Withinia root extract protected and then after treating the the cells with the Withinia root extract for 24 hours, then now you apply the 6-hydroxydopamine, and you can see that the Withinia extract managed to protect the cells from, you can see from the difference in the pathology. And this also has a dose response activity. Next. So this one, they were looking at the levels of ATP in post-treatment. Again, post-treatment in this respect, the cells were first of all treated with 6-hydroxydopamine, and then post-treated with the root extract. And we can see that the one that had the 6-hydroxydopamine is in the middle, and then the one that was having both 6-hydroxydopamine and the root extract has increased ATP levels, increase in about 20% increase in the levels of ATP. Next. So in this paper, in terms of their discussion and conclusion, they were able to show that KSM66 Withinia somnifera root extract had an ability to increase the levels of glutathione in the cell lines. They were able to demonstrate that. They were able to show, I haven't given you all the results, I've just said that, but they were able to show that, again, the extract was able to increase the viability of oxidase and thiol transferase enzymes, inhibit enzyme activities. It was also able to show that the extract was able to modulate the expression of oxidative stress in these cells. Okay, they were able to demonstrate that by measuring the levels of stress response proteins, specific stress response proteins that they were able to measure. They were able to demonstrate that the root extract was able to have an impact on the expression of these stress response proteins. Then there was increase in intercellular ATP levels, meaning the cells were able to improve in cellular respiration. And also there was modulation of redox regulation by decreasing glutathione-related protein levels. Okay, so these results show us very clearly that the root extract of Withinia somnifera has various activities that are able to intervene in the pathology of Parkinson's disease, as demonstrated in this study. Next. Okay, the last hub I want to talk about, or the phytochemical I want to talk about, is cannabis sativa. Again, that is the plant, how it looks like. Somebody has smoked it already. Yes. Unfortunately, I didn't carry any, but looking at the forest here, probably we may be able to find a weed or two. Okay, so that's how it looks like in real life. Next. So this is a picture of the two major active principles in cannabis sativa. That is the delta-9 tetrahydrocannabinol and cannabidiol. Next. So I've just put this picture here to show that the endocannabinoid system that is the endocannabinoid system in the CNS, the normal one in our brains, has a very close relationship with Parkinson's disease and other neurodegenerative disorders. So apart from PD, you have MS, and you also have AD. So that corner there, we have the PD and how it is associated with the endocannabinoid system in the brain, meaning that cannabinoids can actually have a role to play in Parkinson's disease. Next. So cannabis as a plant has shown promise for the symptomatic treatment of Parkinson's disease, but I'm not going to go much into that. I decided just to briefly look at the challenges associated with the plant. As you are all aware that one can actually use the whole plant as in holistic treatment, or one can attempt to isolate active principles. Now, in cannabis sativa, it has been shown that when you go to the laboratory and isolate the active principles, you do not get similar effects as if when you are using the total plant. Okay, and this is a phenomenon that is very common in natural products, particularly the plants, because of the synergistic effect of the various active principles, that when you isolate them, then you lose that synergistic effect. So, but then using the whole plant also comes with its own challenges, and some of these challenges include safety. How consistent can you be when you're using a whole array of active principles? You have the regulatory issues, you have the psychoactivity. If you do not remove the THC, then you have psychoactivity inside. Then you have the side effects, the drug interactions. You have formulation challenges. How do you ensure quality? How do you ensure that you have the right dosage make the right dosage of the concoction? And how do you ensure stability? So all these come into play when you are trying to use a herbal plant for treatment, and particularly if you have to avoid using isolated compounds. Okay. So therefore, the angle that I want to emphasize today is the fact that there is need, if we do not want to isolate, because there are certain active principles that have been isolated from cannabis. There are even medical cannabis, some of them which have been isolated and patented and are being used, but we still have a place for using a total mixture. So the mixture, the challenges with the mixture is that then how do you get to use the mixture of so many compounds? So there's been work that has been done, sponsored by pharmaceutical industries, to try, and actually they have patented a mixture that contains 10 active principles. As opposed to, cannabis contains more than 100 active principles, but one of the pharmaceutical industries has came up with a herbal mixture a cannabis mixture that contains 10 active principles. Now, the research that I want to present here was still say that these 10 active principles are still too many. Is it possible to reduce them to a fewer numbers and still have what we are going to call in this study minimum essential mixture of cannabis sativa that can overcome those challenges that are put there in red. So there is need to identify a subset of components which are responsible for the therapeutic effect, but does not have to contain all those many more than 100 active principles. Okay? Of course, taking into consideration what I've just said, that cannabis-derived ingredients act synergistically. So you don't want to lose the entourage, what we are terming the entourage effect of the active principles acting synergistically. Okay? So this is you can show the study next. So I'll take you through this study, which set out to identify minimum essential therapeutic mixtures from cannabis plant extracts by screening in cell and animal models of Parkinson's disease. Next. So this study came out just this year, at the beginning of October. So it's a very recent study, and the reasons I'm presenting these studies instead of just going through plants is the fact that is to stimulate the students, particularly my students sitting there, to see how you can escalate your pharmacology to these standards. Okay. So the objectives of these studies was to introduce the number of compounds in the patented, as I just told you, a pharma had already patented a mixture with 10 active bioactives from such cannabis sativa. And this study set out to try and see if they can reduce the number of active principles and find the minimum essential mixture that could retain most of the activity, but still contain fewer bioactives. Okay? So, and also to generate, have mixtures that can be pharmaceutically friendly in terms of manufacture, dosage, quality, stability. Okay? So that was the aim of this study. Next. So the methodology is that they set up in vivo neuroprotection assays to assess the ability of the compound. They use the NPP plus, the assay-based cytotoxicity. And then they also did in vivo dopamine release assay, where they were going to see whether cannabinoid and terpene compounds were tested alone or in mixtures to determine their effect on dopamine release from, again, these cell lines. And then they were able to also set up the zebrafish studies to see the effect of these mixtures on zebrafish larvae. Next. So I will not waste a lot of time going to the deep analysis of the studies because the paper is available, recently published at the beginning of October 2022. So just to show, they were able to show briefly, that cannabinoids were neuroprotective, and you can see from that they have a list of the cannabinoids that they were using to applied to the cells, and they were able to show neuroprotective effect. Next, they were also able to show that these cannabinoids, that the ones they had chosen to study, were able to elicit dopamine secretory effect, again from the cell lines they had set up. Next. So to finish, the summary of the results, they were able to identify the cannabinoids responsible for the neuroprotective effect. They were also able to identify the cannabinoids that were responsible for the release, dopamine release. They were also able to identify the minimum essential mixtures containing equal parts of the three. They had identified 3 cannabinoids that they wanted to make sure that the mixtures that they wanted to formulate contained those 3 major cannabinoids. So they were able to identify the minimum essential mixtures containing equal parts of the 3 cannabinoids that were able to alleviate the 6-hydroxydopamine motor symptoms. And within the study, they were able to find 63 additional variations, and then they were able to reduce this to 62 of the cannabinoid ratios. And using zebrafish assays, they were able to come down to 22 cannabinoid ratio variations. And 5 out of the 22 minimum essential mixtures outperformed the regional mixtures, and they are hoping that these 5 that they were able to zero in on can be taken forward for further studies, particularly with respect to applying them in the treatment of Parkinson's disease. Okay, next. So in conclusion, is that cannabis has therapeutic promise in Parkinson's disease, but we need to move beyond the use of the whole plant, but not to lose the entourage effect, the synergistic effect, is an attempt to come up with mixtures. So instead of going to the direction of isolating the active principles and losing the synergy, there is that attempt which is promoted by this publication that you can get yourself these essential, minimum essential mixtures that can be used and can eventually go into pharmaceutical production. So we have a stepwise strategic approach to reduce the complexity of secondary metabolites. And again, this paper is able to demonstrate the techniques that one can use in this kind of study in terms of in silico, in vitro, and they also use medium throughput in vivo experimental systems, and therefore it is important to identify that and see if some of these techniques can be used in our own laboratories, here in our universities and in our research institutions. So next. Good. So that is the end of the presentation, except for the fact that as I was reading through this work, I came across next. I came across this call. Maybe Dr. Gala can have a look at it. I came across this call from NIH, which is focused on cannabis sativa, and is looking for proposals which are involved in promoting mechanistic research on therapeutic and other biological properties of minor cannabinoids and terpenes. And therefore, it is something that as a group we can consider. The deadline, the expiry date is still quite far, so one can purpose to have a look at it and see if it is something. So those when I started talking about cannabis, there was a lot of excitement. So that excitement should translate, as researchers, should translate to coming up with something for our patients. From our locally grown cannabis sativa from our mountains. Okay, next. So what the National Institute of Neurological Disorders and Stroke put out there is that they are accepting research grant applications in the study of minor cannabinoids and terpenes from the cannabis plant in relation to neurological disorders, including but not restricted to pain, migraine, headache, epilepsy, and neurological disorders, which includes our good friend, the Parkinson's disease. And therefore, it is something we can look at. And with that, I will finish my lecture there. Thank you very much.
Thank you, Dr. Margaret, for that very profound and informative talk on the herbal and pharmacological approaches to these neurodegenerative diseases. And I'm sure there must be questions from the audience. So we'll take a few questions before we move on to the next session.
Thank you, Dr. Margaret. That was a very calm and articulated presentation. And the take-home message is that cannabis has therapeutic effect and it can be used for to treat Parkinson's disease. That's the take-home message. So my concern is that it's still like people are exploring this plant, and they are trying to even check its anti-cancer effect and so on and so forth. For instance, someone who is having symptoms of Parkinson's disease, and what the person is looking for is the cure, immediate cure. And if somebody, maybe out of curiosity, somebody just say, "Oh, can you take cannabis?" I was at a conference and they said that cannabis can cure Parkinson's disease. How would the person approach such dosage, taking it, and would there be some side effects in terms of educating people how to use this plant responsibly?
Okay, so coming from the pharmacy world, being a pharmacist myself, the first thing to say is that cannabis is a drug under very strict control. Okay, internationally and also here in Kenya. So its use is not something you just wake up and say, now I came from the conference and Dr. Lucas said you can use, you can use. I'm having some feeling that I need to use, you can't. Okay? So it is highly regulated. I think in most African countries it is still regulated, right from the UN drug control and also in our country. So it's not available for conventional use. It is only available under illicit use. So that is the best, that is the most important thing to note. So actually, me and some of my colleagues have been thinking of how to approach this, and we are trying to write something to see, to take to government, because for it to be released for even now, nobody can accept to tell you that I have cannabis, even for you to take to the lab, okay? Because they know that they'll be arrested. So even for us to use it here at IPR on primates, we cannot until that there's some permissions that have been given by the regulatory authorities and the police in that section. So that is their position. So that needs to be done at very high level to give us access to for research to take place before it can then be used by wananchi. Yeah.
Prof, for this wonderful presentation. I'm just wondering, of course, thinking about much of the control is around the smoking of this plant. And I'm just thinking whether if we worked around the formulations, I don't know whether there are other means possibly we could, apply this drug. Maybe, for example, taking it in tea, in terms of, for therapeutic purposes, rather than the habit of smoking it, because that's, I think, probably where there is much control. But also, of course, one of the things, when I was in secondary school, and we had this young man, and I think he was told that, cannabis, smoking some cannabis will enhance his absorption of. So before his exams for A level, he resorted to, using this, and it wasn't long, and we of course, we witnessed him running around the school, bare chested, throwing off his shirt. So there seems to be a genetic predilection to mental illness issuing from the use of cannabis. And frankly speaking, I don't know whether not everybody really can smoke this drug comfortably. And I think those are some of the interesting aspects to look at. But of course, the other thing is we've been gambling with this issue of the fact that we don't see a lot of Parkinson's disease in our settings. Is it because we have a lot of these, some of these plants available? And probably there are more plants that have protective effects. I don't know. I'm just trying to think around this. But thank you. The presentation is very exciting. Been communicating with a friend of mine whose father actually has PD, and we were joking about how we are going to start his father smoking some cannabis.
Thank you, Prof, for your presentation. My question is, how do we find the balance concerning the cannabis effect? There are so many papers that have reported the psychological, the psychological effect of cannabis. Even there have been reports that says it has some neurodegenerative effect as well. There have been some studies that have been done in our lab concerning cannabis, its effects on some areas of the brain. When it comes to epilepsy, there have been some studies that have been done, especially on the non-psychoactive component of it, which is cannabidiol. Now, how do we find the balance between so many reports that have said a lot about its effects in terms of the positive side and also so many negative side of it? Thank you.
Okay, so I'm glad to hear that there's a lot. The cannabis is apart from Britannia. You're not interested in Britannia. You're interested in the other foods. Yeah. A lot of cannabis is eliciting a lot of excitement. So starting with what you said at the back there, and generally, cannabis contains more than 100 active principles. Okay. So it is really, really a rich source of active that is yet to be studied. So what you're saying, the good aspects and the bad aspects are all resident in these many active principles. So the two most commonly known active principles that I showed in the presentation, the 9-THC, that is the non-psychoactive, abundantly psychoactive aspect. So the people who have made medical cannabis, they have removed the 9-THC from that and made a preparation that does not contain, and therefore they will not have psychoactive effects. Okay, so that it also responds to the guy behind there talking about tea. You can only use it in tea if you'll be able to remove the psychoactive principles and remain with what could be, which could be maybe the part that is not going to have deleterious effects of people running around in the homesteads. So that is quite challenging because it will involve quite a lot of laboratory work. Somebody has to invest in harvesting the plant, removing the psychoactive active principles and remaining with parts that can now be made into tea. So I'm not sure whether that will be possible because if you dry the leaves or the roots and remove the 9-THC, how will it then become tea? It will be quite challenging how to formulate that tea. But what I'm saying is that there's plenty, if you just Google cannabis and its uses in all those diseases that are listed there, that are associated with the endocannabinoid system. There's quite a lot: epilepsy, pain, obesity, stroke, all those things you can use cannabis. Therefore, it is really a challenge thrown back to us to start the research and see how far it can go, depending on your focus. Is it PD, or is it epilepsy, or is it another Alzheimer's? It can be any of those. Okay. But the pharmacology is very exciting for those who want to just deal with the plant and the activities.
Sorry, unfortunately, we cannot take any more questions because we are running behind in terms of time. But I'm sure there will be opportunity to delve into this further during the discussions. So please keep your burning questions. You can even deliberate over this over tea, which is coming soon. She's suggesting they can add a little cannabis in the tea and see what happens to us. So without wasting any time, we'll now load the next talk by Dr. Chuza Huka. Dr. Chuza Huka is a neurologist from here in Kenya, but I suppose he's going to introduce himself.
Hello, how are you? Thank you so very much for inviting me to talk today. It is my absolute pleasure. I would have been delighted to be with you all physically, but unfortunately, I have actually injured myself, and I won't be able to be with you physically. But I'm really delighted to be here, and I hope you can hear me well and see the slides.
Yes.
Excellent. Okay, so I'm a neurologist. I'm Kenyan, born in Mombasa. I live here in Nairobi. I worked at the Aga Khan University Hospital, and we work together with other colleagues in the neurology section there. I'm in private practice. My area of interest and training is movement disorders, and I am also involved in teaching on the first neurology training program in this country. So it's really my pleasure, as I said, to be here. And I'm going to talk about treatments for Parkinson's disease. And really, if I had to rephrase this, then it's all about pills, patches, potions, and probes, really. I am a member of the Africa Education Committee of the International Parkinson's and Movement Disorders Society. And apart from that, don't have any other adverse disclosures. So in these next half an hour or so, my aim is to consider the availability of and access to treatments in Africa and in our country, discuss the treatment options for Parkinson's disease, and outline an approach to real patient management. And from listening to the questions and from just a feel, I get a feeling that the majority of you are now clinicians. Is that correct? Okay, not to worry, not to worry. So Parkinson's disease was described over 200 years ago by a gentleman called James Parkinson, while he was observing patients and people walking in the streets in London, in a part of London known as Shoreditch. But there have been these ancient descriptions of Parkinson's disease, and interestingly, there are some descriptions of Parkinsonism in ancient Egyptian as well as in ancient Indian literature. So the disease has been around for a long time. We get a feeling that it's increasing in prevalence, and I've realized that over the last couple of days, we haven't really seen any patients or any patient videos. So apart from the ataxias, and this is an example of a patient with Parkinson's disease with a classic tremor. And the other real signature of Parkinsonism is what we call akinesia. Akinesia basically means slowness in movement. And this is demonstrated here. You see as he's tapping his fingers, there's a decrement in the motion and slowing down. the other common complement to Parkinsonism is rigidity, and as you can see, there's a bit of a thumb tremor in this patient, but just trying to move his wrist is so difficult, and this is being done passively by me because there is rigidity in this patient's wrist. And often as they walk, they get a tremor. There's a reduced arm swing, as you can see. The arm on the right is hardly moving while the left one is moving. And Parkinson's disease is often asymmetric. Sometimes the tremor is not in the hand, but in the foot. And sometimes also the movement abnormality, the akinesia is in the foot. Just compare that right foot, which is relatively good, to this left foot, and you can quickly see he slows down, has interruptions in movement and decrements in the movement. And this really is what we see in Parkinson's disease. Throughout Africa and in the poorer parts of the world, there's a difficulty in accessing medications for Parkinson's disease, and we have that problem too in our country. And you can see that our rates of medication availability are relatively low as compared to, say, Europe or North America, and even as on a global average. And this has been looked at in Kenya by Jolene Mokaya, who's, I think, done some really good work on this. And there are issues of affordability, availability, obviously adherence, knowledge, and obviously beliefs about whether this medication would work or not, which hindered access to medications more in the public sector than in the private sector, as you can see from this graph there. And this has been looked at Africa-wide, but availability of therapies and services for Parkinson's disease. And clearly, we have problems, and you've seen this picture from Richard Walker's talk. We have problems with access, and as you can see, large parts of Africa, medication is, if it is available, it's not always available, and in some situations, only occasionally available. And it's not just medications. The availability of support groups, for example, we are lucky to have a support group, and some members of the support group, I believe, are logged into this talk. The specialist nurses are hardly ever available. Specialist clinics are not there. Specialist physical therapists are difficult to find, and a whole slew of drugs that we use to treat various symptoms are difficult to find. Parkinson's disease, even though it manifests with these symptoms of tremor, rigidity, akinesia, which is the slowness of movement, it starts long before these symptoms happen. And it's thought that Parkinson's disease begins up to 20 years before symptoms begin, and some prodromal symptoms such as constipation, changes in smell or loss of smell, even depression, sleep disturbance, and a particular sleep disturbance known as REM, sleep behavior disturbance, are known prodromal symptoms for Parkinson's. Now, that doesn't mean everybody who gets constipated will get it, but it does mean that a number of people who develop Parkinson's disease, if you ask them, they've been constipated for many years. And so during this time, there are lots of changes taking place in the brain, particularly in the area of the brain known as the striatum, which includes the putamen, the globus pallidus, and other subcortical structures, including the substantia nigra, where the main pathology of loss of dopaminergic neurons resulting in reduced dopaminergic neurotransmission exists. And obviously, this is where we can intervene in terms of treatment. We can provide more dopamine, or reduce dopaminergic metabolism so that more dopamine is available, and and sometimes you can modify the cholinergic pathway. And these are really the mainstays of treatment for Parkinson's disease. And this is a little bit more detail of the chemistry. And really, when we provide levodopa, which is the main drug used for treatment, it is converted by this enzyme into dopamine in brain cells. And so the discovery of levodopa, the discovery obviously initially of the dopaminergic deficit and the discovery of levodopa was life-changing for people living with Parkinson's disease. And this drug called Cinemet, which is the brand name, but obviously there are various brands, this it changed the life of people with Parkinson's disease. The addition of carbidopa to levodopa simply prevents its metabolism in the body so that more of it is available to brain cells. And really, it improved dramatically the quality of life and indeed life expectancy for people living with Parkinson's disease. And as I discussed with you, the levodopa gets converted to dopamine by dopamine carboxylase, and then is available for dopaminergic transmission at synapses in the striatum or basal ganglia. But the problem is that these medicines, even though it's extremely effective in over 90% of people with Parkinson's disease, initially it's very effective at relatively low frequency dosing, like as little as once a day, but often those like 3 to 4 times a day. But later on, what happens is the effect, the kind of therapeutic window narrows, and instead of it being effective all through the time, there are times when one goes off and the medicine isn't working very well, and when it is working, one often gets abnormal movements, which are rather dancing-like movements, which are known as dyskinesias. And these become more and more prominent as the disease advances, and the off times become greater, and this is known as motor fluctuations. And this is really the problem that one faces with long-term levodopa use, and a lot of effort is now being made to try and address this, the downsides of levodopa use over a period of time. And so people have developed extended release levodopa preparations such as Rytary, which is not available in Kenya. Standard levodopa is available in Kenya. We have the gel infusions through the tube which goes into the duodenum, which is the first part of the intestine, and then there is an inhalation preparation which has just recently become available. But all these things are not available at the moment in our country. But let's hope that courses like this will help increase the availability of these medications as people become more aware and more able to access them. And you've already heard from our excellent speakers before about nuclear and its utility in treating Parkinson's disease, and this is a locally available herb, and there is good evidence, preliminary evidence, that it would work, both from the previous study by a release group from London and from an Italian group led by Roberto Cilli. It's actually a multi-country group led by Roberto Cilli. And there were ongoing larger studies to try and answer this question conclusively so that it can actually be visibly used and obviously reduce the cost of treatment. And then there are attempts to kind of repurpose levodopa, so try another prodrug of levodopa called post-levodopa, which post-carbidopa as a continuous infusion to get rid of these peaks and troughs, especially in advanced Parkinson's disease. And there's also standard liquid levodopa, carbidopa, continuous infusion being tried, but at the moment they're not clinically available anywhere in the world. Sometimes instead of targeting more dopamine in the brain, you can cheat the brain cell receptors into thinking that there is no more dopamine around by using decoy-like molecules. These are the whole group of dopamine agonists. And at the moment, we have bromipexole available. Propinerol is not available. There is a rotigotine patch, which I think is very useful, but is not available. And there is apomorphine, which comes both now as a sublingual spray for, fast-acting kind of for off period, and apomorphine injection, a subcutaneous injection, rather like an insulin injection, and then more prolonged infusion, which we talked about. There are other drugs, like I said, we can cut off metabolism of dopamine by attacking the monoamine oxidase pathway, and these inhibitors of monoamine oxidase B, such as rasagiline, which is difficult to get but sometimes available, and solagiline, which is widely available and cheap in Kenya, and newer drug called safinamide. And these drugs can be very useful, especially in mild early Parkinson's disease and in prolonging the effect of levodopa. And the same for the chemical or methyltransferase inhibitors. Again, they simply prolong the effect of the levodopa, and they allow more dopamine to be available in the brain for longer. These drugs are generally difficult to get in Kenya, but hopefully they'll become increasingly available and cost effective. One drug I do want to talk about is triacetanidil, or what is known as benflexol or Artane. This is a very useful drug for people with relatively young onset, tremor-predominant Parkinson's disease. It is available and very cheap in Kenya, and it has a great role in that situation, but in older patients it causes a lot of side effects, including cognitive side effects, and it's not very useful in that situation. Amantadine is another medication that can be useful, especially in helping with reducing the dyskinesias, which are a side effect of the levodopa. But again, it's a bit difficult to get. The others are much more difficult to access. But another thing to remember is Parkinson's disease is not just a motor disease, it's not just a movement problem, it's not just a tremor, it's just slowing or rigidity. It has many other symptoms. People can develop cognitive impairment and eventually demand. Patients often experience depression at some point. Some patients develop neuropsychiatric symptoms. We talked about the sleep disorder, constipation. People get dizzy because their blood pressures fall when they stand up. They may drool. Fatigue is very common, and so they all erectile dysfunction is another, or sexual dysfunction is another very common problem. And so all these issues need to be addressed, and there are various agents which one can use, some of which are available, some of which are not. But we do have a large armamentarium of agents that are available. And I told you we'll talk about apomorphine infusion. So in patients who have advanced Parkinson's disease, as the disease progresses, and as you've heard, this is a progressive disease, and when simple strategies are not working, one option is apomorphine subcutaneous infusion, so like a pump. Unfortunately, it's not available. It's exceedingly expensive, but it is worthwhile, and there is trial evidence to support its use. And as you can see, there's a definite benefit with apomorphine as compared to placebo in multiple measures, and many patients feel better in a global scale of improvement. The other technology that we do not have available in Kenya at the moment is placement of deep brain stimulator systems into the brain. These are fantastic technology that is again very expensive. I mean, to just give you a feel, the cost of this kind of implant would be something in the region of 5 to 6 million shillings. and it requires long-term use, follow-up. Batteries need to be changed sometimes. With the new devices, you have rechargeable batteries, and the batteries get stuck here somewhere in the upper chest, rather like a pacemaker, and these electrodes are placed deep into the brain. It's all under the skin. Nobody can even see them, and you walk around with them quite normally. Um, there's one advantage with this. I mean, you can tell the security people at the airport that you have metal inside you and that kind of stuff. Um, and these multiple studies have shown to be very useful, and now there's increasing use in earlier disease and in younger patients especially. And there are new targets now for the deep brain stimulation. So the classic targets have been the subthalamic nucleus, so the subthalamic nucleus there and the global there. But more recently, there's been interest in the substantia nigra itself, the one-time nucleus, more cortical motor areas, and interestingly, the spinal cord, and there are a few other targets as well that have been explored. One thing that the arrival of Cuban doctors reminded me of is their extensive experience with lesioning of the brain. So in deep brain stimulation, you put in an electrode, and the electrode gives current here. But in lesioning, you make a lesion with a probe, and then it stays, so it's permanent. It cannot usually be done in both sides, but it's usually done unilaterally. And it does work. The only problem is that there's a greater risk of side effects. A greater risk of side effects, and
Working on that.
sorry, I think my thing is playing on. So there's a greater risk of side effects, and there is also a great lack of reversibility of this procedure. But a more kind of refined way of doing this is instead of putting probes in to make the lesion, you can do the lesion using MRI guided ultrasound. And that is an area of active research at the moment. There are obviously the same problems of adverse effects with bilateral lesioning, but there may be a place in people who have mainly unilateral symptoms for unilateral lesioning. And as you can see, for focused ultrasound and lesioning, you can only do unilateral therapy. But deep brain stimulation does have a great place, just very expensive and at the moment not available in the country. The other more advanced therapy is where you have a pump connected to a tube into the first part of the intestine, that's the stomach. So there's a wall into the tummy, and there's a tube that goes in, it's connected to a pump, and medicine is delivered directly in a continuous infusion. Again, pretty expensive stuff, but useful in advanced disease. I just want to switch gears to other kind of research that is taking place to try and look for more disease-modifying treatments. And this is a glucagon-like receptor 1 or GLP-1 agonist called exenatide. And its research is suggesting that it may have a role as disease-modifying therapy in Parkinson's disease, and there is now a large trial going on, certainly in the UK, and it's an international collaboration. So that's another watch this space kind of thing. And then imaging suggests that it has actually a biological effect in this reaction. So, in summary, if you make a diagnosis of Parkinson's disease, they have little motor impairment, you choose not to treat them or just use exercise and perhaps one of them monoamine oxidase inhibitors. If you're young or with mild symptoms, you could consider anticholinergics. Certainly with any significant troublesome problems or in elderly people, they would go by the answer, and then as time goes on, you often have to add in other medications. So most of these patients would be on multiple medications, and eventually one would have to look at the more advanced device therapies and infusion therapies for them as the disease progresses over the years. And this is another schema to just guide initial medical therapy can be with all these in young patients with tremor only. Anticholinergics do have a place, and then one adds on other therapies, and then finally advanced therapies. But at all stages, exercise has a great role. It's probably the one thing that is disease modifying, and together with physical therapy, as Professor Walker showed you, can help aid and have almost a tablet-like effect. Same for speech therapy, the particular type of speech therapy called loud therapy. It helps people project their voice and communicate better, and it also has a motor benefit. Then there are some other novel things. As you know, with COVID, and even before COVID, people have been looking at new technologies and looking at what are known as virtual households. And so patients coming to the clinic, can you look after patients in their homes without having to go there physically? And it's quite an innovative approach which has been used extensively during these COVID years in the last couple of years. And there is strong evidence to suggest that it's a good idea, and we're beginning to do that even in Canada. With the use of artificial intelligence systems, there may be an easier way to make a diagnosis of Parkinson's disease, but low-tech ways to smell for Parkinson's disease. And there are some people who can actually, with their sense of smell, tell that this person has Parkinson's disease. And people are researching that to see if the chemicals that they smell for can be identified and used as a screening swab test for Parkinson's disease. Because at the moment, Parkinson's disease is largely a clinical diagnosis. And there are lots of artificial intelligence approaches to try and look for new treatments using large throughput systems with available medicines to see if any of them could be used. There are, obviously, with greater understanding of cell biology, attempts to try and attack the actual cellular pathogenesis of Parkinson's disease. Most of these attempts have so far ended in failure, as you can see from all these trials. They haven't really measured up. But there is active work going on in this area, and one promising agent is examitase. There are stem cell therapies trials being conducted in the US, in Europe, in the UK, in the Far East. And these may have a role at some point. These trials, I mean, people have been interested in stem cells from the early 80s, but it's been a long journey with that, so not yet there. And the other thing is because of the central role of alpha-synuclein in the inclusion bodies that are found in neurons in the substantia nigra in patients with Parkinson's disease, people have looked at the monoclonal antibodies raised against alpha-synuclein, and these are the first two studies published this year in the New England Journal in August, looking at two molecules, cinpanemab and with no benefit in a one-year study, with a 2-year follow-up, perhaps too short a study, and similarly with another molecule, prasinezumab, again, no discernible benefit, but again, perhaps the main criticism of these studies is that perhaps you have to intervene perhaps much earlier in the disease, long before it becomes symptomatic, and secondly, perhaps need to be much longer follow-up. So, even though this was the editorial headline, as Churchill once said, the quote this in this editorial success consists of going from failure to failure without loss of enthusiasm. Just hot off the press this morning, this publication, simvastatin, which is an anti-cholesterol tablet to try and modify disease in Parkinson's disease. Unfortunately, no benefit. And that kind of things, the statin story, which has been my point of discussion for a few years. So the future will mean attacking this disease at this point, which means getting biomarkers that can allow us to find the disease very, very early on, even before this prodromal phase. And that's where many of you guys who are into basic neuroscience will be, I think, the heroes. And really, really, that's where focus is at the moment. If we can get good biomarkers for early Parkinson's disease, perhaps even pre-symptomatic Parkinson's disease, and intervene at that time and prevent or delay the development of symptomatic disease. And that's really where the trail leads. But one thing to remember is Parkinson's disease occurs in people. People have other conditions like high blood pressure, diabetes, osteoporosis. They can fall and break their bones. Cerebrovascular disease, other medical conditions. And we talked about the motor symptoms, but also the non-motor symptoms. So the patient is a whole mixed bag of problems that need to be addressed in a holistic manner. And in spite of all the problems, we are all human beings, and we have our joys and our sad moments, and we need to address all these in one person. And I would leave you with that thought as I thank you very, very much for listening to me. And I hope we can have the discussion and experience. I hope this has been useful for you. And again, I want to thank especially Dr. Gulani, who's been so gracious and so kind, and all the organizers, and my immense gratitude to you all, and I wish you all the best. Thank you so much.
Thank you very much, Dr. Hooker, for that very elaborate coverage of the treatment approaches, therapeutic approaches to Parkinson's. We're very grateful for that. And we have, because of time constraints, we'll take only one question before we move on to the next session. Any burning question?
Thank you very much, Dr. Hooper, for a very nice rundown of the treatment options for PD. My name is Abubakar. I'm from Malaysia. My question is regarding the last final slides on the RCT of Symphonimab and Prasinimab, which are monoclonal antibodies that you mentioned probably have not been so successful. If you remember, for Alzheimer's disease, same MAB were introduced, one last year and another one this year. Seems to be a bit more with good prospect. So why is it for PD it's not been so wonderful in terms of
Yeah, so there may be many reasons. I mean, so I think there have been many questions raised about the whole alpha-synuclein monoclonal antibody situation. The first is that the pharmacodynamics is not well studied yet. And as a matter of fact, there are questions about whether the intravenous dosing of the monoclonal antibodies actually reaches the brain, and so impacts on alpha-synuclein in the central nervous system. And even with Alzheimer's disease, the monoclonal antibodies have not been remarkably successful. Indeed, the one that is currently registered is called aducanumab. It's against beta amyloid, it it has a, I mean, even the panel couldn't agree. It was eventually registered that three members of the panel, the FDA panel in the US, resigned because they couldn't agree with that decision. And it's been a controversial decision. I mean, the neurology community is 50-50 split about its use. And even Medicaid in the United States will not pay for it except if it's within a research program. So there is a lot of controversy. There is a newer one which shows more promise, but I think we are quite in the early days of monoclonal antibodies, and I suspect they will have a role that we will find something that we can use, but I think we're not quite there yet.
Thank you very much, Dr. Hooker. And with that, we'll move on to the next presentation by Dr. Mohammed Zahir. Go ahead.
Thank you, and good morning once again. I hope you can hear me. Can someone just confirm?
Yes, we can.
Okay, thank you. So in my talk today, I'll be talking briefly about pharmacogenomics in Africa, and the question I will leave you all with today is: are we really there? And this story, in this particular presentation, I'll be talking mainly about. So those of you who are clinicians, you probably will enjoy this more. I'll talk a bit about human genetics, the introduction of human genetics. I'll talk about the Human Genome Project. We'll look at some of the genetic variation in African populations. We'll look at how underrepresentation of colored populations can be an issue. And then I'll jump right into the topic, talking about the pharmacogenomics in Africa and some of the examples where pharmacogenomics can be made of use, and some of the challenges that we face in the continent and how do we think we should go forward. So starting off with just an introduction to what human genetics. I think everybody probably knows that the basic central dogma of life is really two processes: the transcription, translation, and this is something whereby the transcription is where a double-stranded DNA is converted into an RNA, and the translation is where the RNA is producing proteins. And in fact, essentially, human beings are made up of proteins. So this central dogma of life becomes very, very critical to understand when we talk about genetic variations in the subsequent slide. So all of us are a product of our parents, our mothers and our fathers, and obviously we have 23 chromosomal pairs in each cell. So what you'll appreciate in this picture is a colored karyotype looking at different chromosomal numbers, including the X chromosome and the Y chromosome, which are sex chromosomes. So we have a pair of that from each parent. And the actual challenge is when the system does not go very well, or rather when there is a change in the system, and that we call it variations in the DNA or mutational changes in the DNA. And many a time these mutations cause diseases. Now, what we know about these variations in the DNA is that there are multiple types. You can have single nucleotide polymorphisms where a simple letter within the DNA sequence, which is made up of four main letters, the A, T, G, and C, and 9-thiamine, guanine, acetine, is that where you have a change in the one specific position, and that we call SNP. We can even have simple sequence repeats, so we have a specific position in the DNA where there are multiple repeats. For example, you can appreciate from the picture here. You can have insertions or deletions, so you have new letters joining in the DNA or some of the letters that have been removed. You can have block substitutions, you can have inversions, copy number variations, segmental duplications, as well as translocations. All these are the basic types of variation that we find in our DNA, and one way or the other, these are associated to diseases at some point. Now, when we talk about understanding genomic information to identify genetic alterations, what we are essentially trying to understand is that you have two groups of people. One that have a condition that you want to investigate, and two, you have those people that don't have the condition, which you would call relatively healthy. And you want to compare the specific region within that DNA where they differ. And when you find that space in the DNA where you have a difference between a patient and a healthy individual, and you have a lot more of those differences between patients and individuals in the same location, that's when geneticists make a conclusion that that particular region might be associated with that particular phenotype. And so that information is compared, and that alteration can be associated with the disease identified. Now, the story of genetics cannot really start without talking about the Human Genome Project. This was basically started by Dr. Francis Collins and Craig Venter, John Craig Venter, in the year 1993, which is essentially the largest biological, international biological collaboration that has been recorded. And the objective here was really to be able to sequence the 3 billion base pairs of DNA that make up the human DNA. And they wanted to understand the arrangement of the DNA, and they wanted to write the entire sequence, which is 3 billion base pairs. And so the project itself had multiple milestones. It started off in 1990, went all the way up to 2003. And in the process, for example, in 1990, the first BRCA gene that is known for causing breast cancer was found. They were able to find, sequences of Saccharomyces cerevisiae, that's the yeast, sequence of the bacterial genome, sequence of the E. coli, sequence of the roundworm, the mouse, and subsequently the fruit fly. So while they were in the pursuit of getting the sequence of human DNA, in the process they also managed to get sequences of various other species. And together that becomes a very informative way for geneticists to understand variation and correlate that to susceptibility for a specific gene. So goals of the Human Genome Project was really to determine the sequence of DNA. We wanted to identify the genes in the human DNA and store that information in databases such that we can have improved tools for analysis. And essentially, we also wanted to address the ethical, legal, and social issues that might arise from the project. Now, I think most of you probably understand that next generation sequencing has been a hallmark for the field of genetics, and that has literally transformed how we talk about genetics and how we talk about precision medicine and the way forward. When the Human Genome Project started in 1990, it took $2.7 billion to actually get the human DNA sequenced. But over time, it has been a transformation step for utilizing next generation sequencing, simply because the chemistry, optics, fluidics, the computational force has subsequently been improving the technology. And so by the time years were passing by, the $2.7 million that was needed to make the sequence of the human DNA now can go up to $5,000 in 2015. And in fact, I just freshly got off from the American Society of Human Genetics conference last week. And in fact, we are down to $10 a genome using the Ultima Genomics platform. So it's not going to be very difficult for us to be able to sequence multiple African populations very, very soon, because cheaper technologies are already in the market. And so we must have to make use of that. And because the prices went down for these technologies, it is very clear that the number of genetic tests that were available on consultative services increased tremendously over time. So you can appreciate from 1993, there was barely about 20 to 30 tests, to 2013, which went up to 2,200 tests. And now these tests are even more than 10,000 numbers. So it's exponentially increasing the number of genetic tests that are available simply because of the way the technology has transformed the costing. So this is a picture of a 23 chromosomes that we have, that I initially shared with you in the first slide. And this is a picture that was taken in 2006, where much of these chromosomal areas are blank, and then these tiny little blobs at specific locations within these chromosomes are areas where we researchers found that there is something, a mutation or a variation that can be associated to a disease. Now watch how this changes over time. In 2006, this is what it looked like. In 2007, more and more blobs were coming up from different chromosomal locations being associated with diseases. In 2008, more and more came up. In 2009, it got even more. And as of 2019, there is not a single place within the chromosome and the DNA position that has not been associated to a disease yet. So you can imagine, due to the technology, due to the costing, how much we can in fact be able to find associations of these genetic positions to susceptibility to disease, and that just tells you the importance of genetics in the health field. Now, why is genetics so important, and what can it do? So apart from telling you about your specific trait in terms of your eye color or your hair color, which is sort of cosmetic in nature, but genetics or genetic information can tell you the differences in response to diet or medication. It can be able to tell you the influence on the likelihood of a disease and whether or not you are carrying a particular mutation that is explanatory to your phenotype. Now, that was from a diagnostic point of view. From a therapeutic point of view, you can imagine that in the normal circumstance, you have a protein and then you have an interacting drug. And when you know the information about the protein and the gene, you can create a drug that would match with the protein. But in the instant where there is a mutation that has changed the gene, and that subsequently has changed, as I mentioned, the central dogma of life, so your transcription and translation is now producing an altered form of protein. You then need that information to create a different drug such that it would interact with the altered protein. So that genetic information is very, very important also for determining the drug-protein interaction to be able to create drugs that will be specific to the variation that you are associating with. So what about the genetic variation in the African population? Because essentially this is one of the areas that is of prime interest. What we know from the Thousand Genome Project is that Africans, which are represented by the yellow blobs on the top, and this is the rest of the population, you can clearly see that the number of variation that's present in the African population is significantly higher than the variations that we find in the rest of the population. So that tells you that the genetic data sets that we currently have do not have specific population information of African populations, but this is where an area that really requires countries to come together to really be able to sequence their populations such that that increases that representation, because these are some of the problems that the Human Genome Project were limited, and this is something we'll talk about in the next couple of slides. But the main message here is that Africans in general have a much higher number of genetic variation compared to the rest of the world. And number two, not only do they have much higher genetic variation, but they are also different from each other as well. So this is a plot that tells you different genetic locations within the genome, and then the specific geography of the African populations where they come from. You can appreciate that within the West, Central, and South Africa, and the multiple tribes, the Xhosa, and the Congo, and the Bong, and the Hausa, have a specific genetic makeup that is similar to each other. But then the same genetic makeup is a bit different from the profile of genetic makeup from those people in East Africa, such as the Maasai, Sudanese, the Gumuz, the Lukia, the Bulala, which is also very different from how the profile of populations in the Horn of Africa, such as Somalia, look like, which is again different from North Africa, West Asia, and South Asia. So we learned two things from the Thousand Genome Project. That number one, not only Africans have a lot more number of variation, number two, they are very different from each other, even within the continent. And so that becomes a very challenging task to be able to find specific variations in the DNA that would explain how disease mechanisms are caused, and how susceptible is a person to a specific disease. This is a picture of a principal component analysis for a genetic structure. And we've divided this into A and B. The first part is global populations, and you can clearly see that sub-Saharan Africans come together, although the Hadza populations of Tanzania are a bit separate from the sub-Saharan Africans, whereas the rest of the continent come together as well. So you have the Western Euro-Asians, the Oceania, the Eastern Asians, and the Native Americans. When we dive into the sub-Saharan African population, you can also clearly see that the sub-principal component analysis will show that East Africans come together, West Africans will come together, Pygmies will come together, and the Hadsas are remaining distinct. And so that tells you that the geography of these populations also has a role in determining how similar or how different these populations are genetically to each other. And this has everything to do with the whole human migration story, which is a very informative story because essentially every one of us, whether we are online or in that room, is an African. And when I say that, that's because Africa is the cradle of humankind. And what we do know is that Homo sapiens, or human beings, originated from Africa roughly about 200,000 years ago, and they spent some of their time coming with the migration out of Africa. And you can clearly see from the picture here that the majority of these people that went towards Europe and Asia came out from the Horn of Africa. And so that means that you can pick up a bottle of Skittles. For those of you who know Skittles, these are basically colored sweets. So red, blue, green, all sorts of colors. And that's sort of the analogy I used to represent African populations. But now, when these populations migrate, only the yellow and blue came out. And so that means that the European populations and Asian populations are from a different color. And when they marry and when they have kids, they are basically making babies out of the blue and the yellow that came out. And that means that there is more similarity within the genetic makeup as opposed to having multiple colored Skittles that are still present in Africa. That causes the diversity. The other issue is also that the continent of Africa has major population groups that can be divided into multiple language families. All right, so we have the Afro-Asiatic language, which is spoken mostly in the north. We have the Nilo-Saharan. We have the Kosa, which is based in the south, and then we have the Niger Kordofanian, which is the sort of the west, central, and east portion of the African continent. And apart from the migration of the population out of Africa, there is also a migration that's happening within the continent. And so you see that multiple arrows that are showing in the continent have shown that populations not only went out, but some of them came back in and then relocated from the east to the south and from the west to the east. And so that creates also a difference in genetic pool, and it creates that diversity of the African populations that we find in genetic data sets at the moment. And this is proven by this particular linear plot, where you can clearly see that the sub-Saharan Africans represented on the left have a far more lower linkage disequilibrium compared to the non-Africans, which have a more shared pattern of LD. And that means that when the population migrated, there was less and less difference between these populations. So the Euro-Asians have less difference within themselves, and then when the Asians came in, they have less differences between themselves, and so on and so forth. Now, what about the Human Genome Project that I was talking about the limitations? So what we know is that the Human Genome Project essentially made a huge blunder when they did not take enough samples from African populations. And that's why you still find most of the recent literature that is still producing new genetic information from African populations that have previously never been recorded in human genome data sets. That means that there is a lot of information that the Human Genome Project did not capture from African populations. And then that subsequently also led to a problem, is that the current population of the globe was highly underrepresented in these genetic data sets, in these genetic studies. Because if you look at even the GWAS studies right now, 97% of that data is purely based on European ancestry, whereas only 3% of that data, in fact, is getting even lower now than is being represented by colored populations. populations, and of that, only about 0.5% is actually African population. So that creates a huge issue for us that are utilizing these kinds of variations to make conclusive diagnoses, especially in the molecular clinics, because I'm not able to find how to classify that particular variant of unknown significance utilizing a European ancestry on an African population simply because of the difference in the genetic makeup as explained earlier. So, after that, a lot of movement took place with African scientists talking about the genetic diversity of sub-Saharan Africa. And obviously, people started saying that, right? The genetic research, Africans are largely underrepresented, and the genome variation across the communities will provide a greater understanding if these populations are studied. And essentially, there is a huge problem with the core of the Human Genome Project because the African population was simply not there. And probably most of you know this is Professor Charles Bakumi. He's also the founder of the African Society of Genetics. And people like him raised voices to ensure that African populations were given considerable platform so that these populations are studied. At the point that the Newsweek even published in their first page something like Black Genes Matter. So how does that have an influence on the pharmacogenomics? And that's what I want to really talk about today. So pharmacogenomics is basically a study of the genetic variation that influence individual response to drug. And basically what it does, it combines the traditional pharmaceutical sciences, like the biochemistry, with really understanding the common DNA variation in the human genome. And for example, this picture tells you that of the multiple drugs that are present in the market, and this is an average, so it's not an absolute number, but these are average numbers, of the drugs that are available in the market, that, for example, we use them for asthma or antidepressants or diabetes, arthritis, not all of them are effective. In fact, if you look at antidepressants, about 38% of the drugs are ineffective, right? If you look at diabetes drugs, about 43% of the drugs are ineffective. In fact, if you look at cancer drugs, sometimes even up to 75% of the drugs are ineffective. And so that tells you that one size really does not fit all. And that means that there is really a huge importance of including African populations or color represented populations to be able to understand how a particular drug would benefit that particular patient. And this is something that we'll see in the next slide. So there is a concept called tailored medicine effect, whereby you have a population group. Each of those different colors represents a different genetic makeup or differences in the genome of that particular patient. Standard approach, normally we basically provide prescription to a specific disease. We give all of them the same drug. But in fact, utilizing that approach, only 20% of them actually benefit, while 80% is just simply waste because those people do not benefit from those drugs. And so ideally, we would want to go towards a more tailored approach whereby each of those groups, once we have identified from the DNA which category of variation they are carrying, subsequently then we'll be able to say which of the drugs will be benefiting these particular groups of patients. And the reason why that happens is because there are certain DNA variations present in these groups whereby the drug can sometimes be toxic and also not beneficial, so you want to avoid giving them those drugs. Sometimes the drug is not toxic and is also beneficial, so you want to give them the drugs. Sometimes the drug is not toxic and also not beneficial, so you want to have a certain dosage requirement for those patients. And sometimes the drug is toxic, but it is also beneficial. So you want to go around certain ways of how you want to treat these particular patients. And because this genetic makeup is so vast and it makes it very, very important for us to understand these variations in terms of the relation with the drug. Now, what really are the factors that affect the variation in the drug response? Particularly when you have a drug, you also want to understand the genetic factor, but at the same time, you want to understand the environmental factors. So that includes the age, lifestyle, diet, and all of those things have an influence on how the drug is absorbed, distributed in the body, how fast is your metabolism, and how fast can you excrete it. Because essentially, drug has to get in the body, get done its job, and then it has to leave. And so the whole drug response will follow these particular factors. What we do know included in majority of them are in the oncology sector. And obviously, you also have some of these pharmacogenetics and pharmacogenomic researches where there are already drug labels for cardiology, neurology, hematology, rheumatology. And so you can imagine that there is a lot of scope to improve diagnostics, particularly for different fields, when pharmacogenomics can be an approach to find out which drugs will actually work and which drugs will actually not work for those patients. What is the situation in Africa? So if you look at the number of pharmacogenetic studies that are found to be conducted within the region, 33% are happening in North Africa. You have about 12% happening in South Africa. You have about 6 to 7% in West Africa and 7.7% in East Africa. And 42% of these are in fact happening in the US, but on African Americans, which is why it was coupled in this particular plot. And so this is by no means comprehensive. And so it tells you that there's a lot more work to be done as far as African continent is concerned. And if you look at the number of pharmacogenetic studies that are within different fields in Africa, this is quite evenly spaced. You see in the blue, cardiovascular disease has about 20%, 18.5%. Diabetes, obesity, cancer, infectious diseases, kidney diseases are some of the main fields within African continent where pharmacogenetics and pharmacogenomics studies actually take place. This is perhaps the most important slide. So pharmacogenetic effects of the genotype in a drug metabolic enzyme. And you can imagine that when you have a patient, you find the diagnosis, and you now want to prescribe a medication, you have a standard dose for that particular group. But imagine if you knew their genotype, and you have 4 different types of that particular genotype, in which in the first group, you have both these alleles are in fact inactive, and that means that these group of patients are actually not going to be able to metabolize the drug. So you probably want to reduce your drug dosage. You have a second group whereby one of the alleles is inactive, the other one is active, and so that there is some chance or some room for that active allele to actually metabolize the drug. And so you want to reduce the drug dosage, but not as much as you would reduce it for those alleles that cannot metabolize. In the circumstance where you have normal alleles, you will maintain your dosage. And in circumstances whereby you have multiple alleles, that means that these patients are actually fast metabolizing. And so you want to maintain probably higher plasma concentration of these drugs. So you want to probably increase the drug dosage. And by getting this kind of information, it is very, very important to determine the pharmacodynamics and the pharmacokinetics of these drugs, which means that what the body does to the drug and what the drug does to the body in correlation to how the dosage and the genotype will actually have an influence. Now, once this information is obtained, you can actually classify them in various categories. And it is very, very important to have a population-specific data. So this table tells you how the particular variants, for example, if you take the CYP2C9 gene, different variants have different frequencies of these variants in different populations. And once you have that information, it tells you pretty much how much of a specific population will be able to benefit from that particular drug or not. So even health systems can take decisions based on the frequencies of these specific variations that tell you whether a particular population will be able to metabolize the drug or not, and whether a particular population will have any benefit or not. And subsequently, this kind of information also will determine the drug dosage. For example, here, this is a 3-range expected maintenance of coumadin, which is basically warfarin. Daily dose is based on the CYP2 C9 and the VKORC1 genes with specific alleles. And when you have a certain genotype, so here in the case of the TT, while you have both the 1-star alleles, you probably want to maintain that 5 to 7 milligrams of the drug. But as soon as that changes from GG to AA, now you see all of a sudden the drug dosage actually is changing. And so this kind of information becomes very, very pertinent, particularly looking at population data or individual data to tell how much of a specific drug can actually be given for treatment. So when we look at the global distribution of some of these phenotypes, so for example, you have the CYP2D6 gene, which is basically the enzyme that's responsible for breakdown of opioids, antidepressants, and multiple other. In fact, 25% of majority of the drugs that we intake are metabolized by this particular enzyme. So that tells you that by charting some of these variation frequencies, where these particular populations were metabolizers, intermediate metabolizers, and ultra-rapid metabolizers. And having that information population-wise or country-specific-wise, it will tell you what kind of dosage to give that particular population. The same can be said for the CYP5119 phenotype that is basically used for the antiplatelet therapy breakdown, and some of the information that can be obtained from such kind of information is whether or not these are poor or rapid metabolizers of these drugs. Now in Africa, we have also the distribution of the 5 inferred CYP3A5 haplotype. This is basically the enzyme that's responsible for breakdown of some of the steroids that we use commonly for liver, kidney, and various other organ damage treatments, as well as the xenobiotics that are being used in the continent. And what you would appreciate, so this is a picture that was created with information from about 2,200 individuals from 32 different and ethnically diverse African populations. And you can clearly see that the GG allele type are much more seen in the northern population, and the East African population has a slightly more increase in the GG compared to the rest of the West and South African population. So that already gives you a lot of information about whether a specific type of drug that's responsible for the breakdown of some of these steroids and xenobiotics, how they will behave in specific populations. And what we know so far from the African data is the pharmacokinetic and the pharmacodynamic gene-drug pairs that has been created by the Clinical Pharmacogenetics Consortium, together with the Dutch Pharmacogenetics Working Group, in which these variants are actionable, right? So of the genes that you see on the right, on the top, the blue ones are all the variants or the genes that are responsible for the metabolism of the drugs that are present in the blue line for the pharmacokinetic studies. And those variations that are found in those genes are in fact clinically actionable. That means that you can already determine drug dosages utilizing which kind of a particular variant this population is getting. And at the same time, at the bottom, you have the red, which means the pharmacodynamic drug pair. And again, the same thing, using that information, you're already able to tell the pharmacodynamics of a particular drug for that particular patient. Now, just to give you some quick examples of how pharmacogenomics can be so influential in the continent, one story that I really like to tell a lot of people is, the typical efavirenz treatment for HIV. A couple of years back, Zimbabwe utilized the efavirenz, so they shifted from a 3-drug cocktail treatment for HIV into a single dosage of the efavirenz, which is sort of, it couples all the 3 drugs into one cocktail. And that was sort of laid out in a trial led by Professor Colin Makingrembwa to look at how these patients that have HIV would in fact react to efavirenz. And what he found was that a lot of patients that were carrying the normal allele in the CYP2D6 were okay, whereas if you have an altered allele in the CYP2D6, then you have a serious side effects, including suicidal tendencies and various other mental disorders, particularly because this particular gene is responsible for the metabolism of that efavirenz drug. And so, in fact, those that are not able to metabolize it properly were in fact getting side effects from the drug over toxicity. The same thing can be talked about when you talk of the ban of codeine in Ethiopia. So in Ethiopia, populations have quite a number of gene duplications in the CYP2D6 gene, and that is causing the change of codeine to morphine in the body. And so subsequently, there's a lot of side effects that was seen in these patients that were taking codeine for treatment of multiple diseases. And so it is important that this information is priorly known so as to prevent fatalities. And so that means that there was, to a point that Ethiopia had to ban codeine for its population once they found out that the population was a high carrier for duplication, the copy number variations for the CYP2D6. The other example is also the breakdown of chloroquine. Basically, we treat malaria using drugs such as chloroquine, but in Senegal, we've seen that specific alleles are present in the CYP2C8 gene that are not able to metabolize the chloroquine properly. And then we see a lot of side effects that we find in those populations that do not have a proper mechanism to metabolize the drug. So these are some of the quick examples that I can share with you that these are all published, so you can easily do them, and you can find information about the variation of these populations. So the question is, are we really there yet? I think main factors that, so at the moment, one of the issues that we have is that clinicians in sub-Saharan Africa do not really report adverse drug reactions, right? And that becomes a huge problem for us in the continent to understand whether or not a particular drug may have a side effect or not. And this is probably because of the lack of competency towards pharmacovigilance, or maybe absence of financial incentives. Maybe there is a belief that only fatal ADR should be reported and not the side effects. Maybe there's a fear of submitting inadequate ADR reports, or a belief that reporting of that ADR adverse drug reaction data would have no consequences, or sometimes just the absence of communication or the drive to publish this in the peer-reviewed journals. So these are some of the reasons that we've seen that has led to clinicians not really reporting adverse drug reactions. But I think this is very, very important that clinicians have to start reporting the adverse drug reactions so that this can be picked up by molecular scientists so that they are able to correlate the genotype with the reactions. But we have both pressing issues in Africa. Particularly, we really need to build research capacity for genomic research. We need to improve medical genetics, not only education but also services. We need to set up genetic testing for congenital diseases, which is at the moment very much lacking. We also have to think about some of the ethical considerations for genomic studies in Africa. And most importantly, how to build a conducive biobank for genomic studies in Africa that takes into account our social and cultural needs. We have to create a standardized biorepository, which is basically a resource for high-quality sample storage and bioinformatics and pharmacogenomic data set for African populations. These are some of the areas that I think are quite pressing in Africa. But again, with challenges, there are also opportunities, and I think there's a lot of work that African scientists have already done. And some of the milestones that I can point out very proudly is the establishment of the Human Health and Hereditary in Africa, so the HP Africa Consortium, that looks to foster the career of researchers and perform research in genomics and genetics to increase the representation of some of these data sets in global genome maps. We also have the Malaria Gen International Haplotype Mapping Project. We have the TOPMed, we have the Pan-African Epimetrics Panel. We also have the Pharmacogenetics Association of Africa and various other organizations that are trying hard to come together to address some of the pressing issues that I mentioned in my earlier slide in such a way that the African populations are not left behind and that genetics and genomics has a role to play when it comes to precision medicine in Africa. What I think are some of the key pillars that describe major aspects when we want to especially catalyze these genomics in a successful manner. One is really to map it, so identify African policies, programs, stakeholders that would be able to be interested in such a study. You then want to capacity build, so you build your bioinformatics structure, you want to build your workforce, you establish training, you want to have a biobank, you want to have a good lab setup. You then exemplify the establishment, so you really build on establishing research collaborations. So this is where Tanzania talks to Kenya, Kenya talks to Uganda, and so on and so forth, whereby intercontinental and as well as outer mechanisms of stakeholder engagement is done. And finally, creating a strong, engaging bridge with stakeholders at a global level, which becomes very, very important for implementation of pharmacogenetics in Africa. And some of the things that where we talked about the barriers of implementing pharmacogenetics, that includes, of course, the under-resourced healthcare system, the challenges in genotyping, social cultural issues, and basically the lack of clinical information. Into a future for implementing pharmacogenetic testing in sub-Saharan Africa is to then create large-scale African genomic initiatives, set up a biobank, find ways of doing clinical pharmacogenetic testing, and train. That is very, very important. So in the end, what would be really ideal is that when there is a patient who is exhibiting a certain sort of symptom, goes to a doctor, the doctor goes to sends the sample, especially the DNA, using a genomic medicine center to a centralized processing facility, which is basically an innovation ecosystem that provides the data back to the doctor, who will then be able to say that a specific patient will be able to benefit from a certain drug or not. And then so that becomes a mechanism to provide specific drugs that will actually benefit the economy. And it's very important at this point to also realize that it is as much important to, I say stakeholders cannot necessarily only be the patients and health professionals, but stakeholders at this point are also government officials. And so it becomes our responsibility to explain to the government in a language that they understand. That's particularly from the economic point of view. Each year, when we do not implement pharmacogenetics and pharmacogenomics, we lose billions of dollars on wasted drugs, on wasted resources that would otherwise be of a different value if utilized appropriately. So it is very, very pertinent that policymakers and stakeholders at that level are also taught about the pharmacogenetics and pharmacogenomics and how best can that be of economic value to some of these African populations. So in summary, pharmacogenetic studies in our population are less abundant as opposed to other populations. Most of these studies that do happen are available for warfarin, but there is also a lack of studies for some diseases such as sickle cell disease, which is something that really needs to take into consideration, given that sickle cell is quite prominent in the continent. And if we look at the infectious diseases, pharmacogenetics at the moment has focused really on HIV and TB, looking at the treatment of efavirenz and rifampicin. We do need disease-specific pharmacogenetic screening panel, and this can certainly be feasible in the future. And more precise medication for African patients can save the healthcare system the costs of the inadequate treatment or the need to treat adverse side effects for our patients. So with that, I really thank you for your attention, and I really thank you for the invitation to be able to speak in this workshop, and I'm happy to take any questions.
Thank you very much, Dr. Mohammed, for that very stimulating and insightful talk. But again, because of time constraints, we're only going to take one question so that we have a little break and come back again. Anyone?
Thank you, Dr. Mohammed. So I thank you for that presentation. So I would like to know that about the actionable drugs, pharmacokinetics actionable drugs. It's been a challenge to actually figure out the most important actionable drugs when it comes to pharmacogenomics. So, and even on a global stage, not to talk of Africa, that we have data deficits to annotate pharmacogenomics at the large scale. So what would be your advice on this?
Thank you so much for that question. If I understood you correctly, what you are essentially asking for is how can we utilize large data sets for the treatment of our patients, utilizing some of those variation data, and how can we maximize usage of these information. I think for a start, there are multiple approaches to this. In fact, if we look at the way how Europe has done it, is looking at specific populations at small scale and then clumping that data into larger data sets with pure comprehensive genotype and phenotype information. And that is something that we also need to do in Africa, whereby we start with a common disease pharmacogenetics panel, whereby we are able to identify specific variation that would be informative for us looking at basically which of these candidates would be a fast, rapid metabolizer of the drug or a slow metabolizer, and then extrapolate that information to multiple countries throughout the continent in a similar method. And that would then be a consolidated data set, which will have much more statistical and clinical significance to say that a certain variation may or may not necessarily work for us. And that becomes a leverage for African governments and African people to go to these companies, their pharma companies that have millions and millions of investment in creating these drugs, that say that this is about time we change how drugs are brought into the continent. Africa should not be a dumping ground for drugs that are produced using data of other populations, but rather we should be in a position that can generate our own data, and that data has to be incorporated into the mechanism of drug production that is specific to the population that we are looking at. That is the true spirit, in a way, that Africa will be able to benefit from precision medicine, because even, as you probably now know, that the drug dosages related to some of these drugs will also change, because it is very pertinent to understand the dynamics and kinetics associated with some of these variations, particularly from the DME point of view. And subsequently, these drug targets will have to have massive changes in how they are being formed, looking at the genetic makeup of our populations. So consolidating of a lot of African data would be the way forward, such that we are identifying specific regions within the genetic makeup that would then be